Okay, so hello everyone, welcome to the Biology Bioinformatics Q&A session. So list of talks are on the website, and so I'm going to go in alphabetical order. Each of the speakers is going to be given about a minute to kind of discuss their presentation, and then we'll have a Q&A session. If you have any questions, feel free to either add them into the chat and I'll add them, or just ask a question. So let's get started. So the first talk is Avocado, Characterizing Human Epigenomics Through Imputation. Jacob Schreiber, right? Mm-hmm. All right, cool. So let's get started.
Yeah, so the main motivation behind this work is that genome scientists will generally try to measure various forms of biochemistry within a cell. Sometimes these are things like, is a gene expressed? Is a gene tightly bound around its histone scaffold and thus likely to be, likely to not interact with other things? And so one of the reason that these measurements are super useful is because they can explain, what's going on in the genome. If you have a gene that's expressed in one cell type and not another cell type, that probably helps explain why, what the molecular difference is between these two cell types. The problem is that we can't perform all of these assays in every single cell type in the body that we'd like to, because there are hundreds and hundreds of different cell types we could profile, and we don't have the time or the money in order to perform all the experiments. So we proposed a deep tensor factorization model named Avocado that is supposed to take all of the existing data that's been collected and to impute the output of the experiments that we'd never be able to perform. And so the idea is that you can impute these experiments, and then you can use them in order to guide the in order to guide future experimental efforts. So what we did is we compared against other efforts in this field to try to impute experiments. We found that not only on a smaller data set did we outperform the previous work by a significant margin, but we found that when we applied it to a much larger data set, which these other methods couldn't be applied to, that we were able to model a ton of different types of activity. For example, transcription factor binding, histone modification, all of these types of things. And we did so in a way that was much more accurate than methods that are trained specifically to predict protein binding. There's another aspect of the talk that I got into a little bit, but isn't on the imputation side, which is the fact that this model ends up also learning low-dimensional latent representations of all of these axes, like of all the cell types in the human body, of all of the different assays, and of every position along the genome. And we found that these are incredibly useful as well. So we're really excited by this work because we found that both the imputations and these low-dimensional representations that we learned are both useful, and both have been able to end up with future work.
Okay, so if no one else has any questions, okay, well, I think I had a miscommunication with Jill or something, so session's supposed to start now. Okay, so here's one question that someone had. So do you have an idea of the uncertainty related to protein binding measurements? Is Avocado having a kind of benchmark there?
I'm not sure exactly what you mean in terms of the uncertainty related to protein binding measurements. We found that we were able to impute protein binding measurements much more accurately than participants in a recent challenge that focused on predicting transcription factor binding. And in this challenge, we didn't use any of the information within the challenge. These were simply the output tracks that our model imputed. But I think that's somewhat related to this other question from Kelvin about uncertainty estimation. So maybe I should answer both of those at the same time.
Oh, yeah. Sure.
The idea is basically that in addition to getting what the prediction is, can you also get a measurement of uncertainty? And so the answer is that in the model that we release in the papers, we don't include a measurement of uncertainty. However, it is possible to do so. That we have trained a model where the idea is that rather than making a point prediction, we're trying to predict the signal at a certain position, you try to predict a normal distribution, where you want the variance to be as tight as possible if you're very certain, or the variance ends up becoming wider when you're less certain. And so what we found is that we got similar performance, but that it was unclear what precisely we could do with such a thing to, write a paper on it. So the short answer is that we haven't included that in the papers that we published, but it is something that we could easily do.
Okay. Okay, so since we kind of are having a little bit the Q&A session, kind of starting confusion and stuff, I'll just say one more question. This one is pretty simple. So avocado, how did you choose the name?
Right. So the reason was that in the same way that when you're cooking, you can substitute in avocado for traditional oils, that you can substitute in the imputations for the output from traditional experiments.
All right, cool. So yeah, I'm sorry about the whole timing thing.
No problem.
If we have any time at the end, I'll come back to you and we can finish doing some more Q&A stuff. But for now, I'm going to invite our next speaker up. So this talk was called Continuous Updating of Evolutionary Estimates in the Open Tree of Life. And so speaker is Luna L. Sanchez Reyes from University of California, Merced. So take it away.
Hello, everybody. Can you see my screen? I guess, right?
Yeah, I can maximize it.
Okay, cool. So basically, what we're trying to do in this Python toolkit that we've been developing in the McTavish lab is trying to solve a question that a lot of evolutionary biologists have been asking for a long time. And it's basically that we want to know the evolutionary relationships among all extinct and extant organisms. But this is a problem of scale. We have more than between 3 million and 100 million living species today, and this represents only 0.1% of all the species that have existed since the beginning of life on Earth. And there are a lot of databases that have been accumulating data that will hopefully allow us to understand these evolutionary relationships. There's the Open Tree of Life project. There are databases that accumulate DNA information, and these databases have been accumulating also a lot of data and analyzing. There is this other database, Treebase, that stores curated molecular data, and all these databases have been there have been pipelines that have been developed to scrape information and data from these databases, but there is not a pipeline that or an easy way to make these databases inform each other to try to answer this question about evolutionary relationships. So this is the main goal or the main motivation for PhyScraper. And it basically integrates data in a way so it can be easily reanalyzed in different ways, so we can update our knowledge on evolutionary relationships. And we chose Python. Well, it is written in Python, and it can be run in a virtual environment or using Conda. It can be run as a command line application and on a Jupyter notebook. Like we have been developing all these platforms, so it is easy to use. And one of the main reasons why we chose Python is because the Python dictionaries are a great way to to allow this interoperability and communication between databases. And then this allows us to write the data in a more human readable way, as like in JSON files or CSV files, so that users can see how the databases connect between each other. And hopefully this will allow for knowledge in evolutionary relationships to grow faster. And we've been working on the documentation, and we have all the code open on GitHub if you want to check it out. And yeah, now time for questions if you have any. Oh, you are muted.
Sorry. So yeah, I had a question. So are there any tools in other programming languages that you know of that do similar things, and are you compatible with the outputs of those tools?
Not really. To our knowledge, this is the first software that does this integration of databases. There are pipelines that scrape GenBank, for example, and molecular databases. Those have been developed a lot, but they do not really connect to the Open Tree of Life or to GBIF, for example, or to Treebase. Yes, I don't think there has been many efforts on that side. It's really hard from the taxonomic viewpoint. It's almost impossible it takes a lot of time to match different taxonomies because each database has their own taxonomy. So we are taking advantage of the Open Tree of Life platform that has been developing a unified taxonomy. So this is what we are using. And yeah, I don't think no other tool has been using that. But the goal is that it could be, the outputs should be able to be used by any other platform. The outputs are basically Newick and Nexus trees.
Okay, awesome, thank you.
Sure.
Okay, so we're going to now invite our next speaker up. So this talk was called Frictionless Data for Reproducible Biology, and the speaker was Lily Winfrey. So if you would like to kind of give everyone...
Sure. Hi, everyone. And it's a pleasure to be at Virtual SciPy. And so the basic idea behind my talk is about the Frictionless Data Project, which is an open source project. And we work with researchers to help them do kind of some of those boring data cleaning tasks. So we do things like help document metadata in a standard way. We help package data so it can be more usable, so it can be published easier, or it can be used between different tools in an easy way. We try and make this process more interoperable by making everything machine readable. And we also have some validation tools. So we have tools where you can create schemas and then use schemas to validate your data. And then I talked about a few use cases for biology. So for example, we're working with the BICO Demo group, which is oceanography researchers, and they actually have a talk later in the afternoon today. And we've been working with them to create a data pipeline where they are inputting raw, messy data about oceans and keeping track of the rich metadata in this machine-readable, interoperable JSON format, and then are able to host that data for other people to use. So the whole point here is that we're trying to make data more usable, more understandable, and make research more reproducible overall. And then I also showed an example from a graduate student where she was interviewing people, which is pretty messy data in general. And then she was able to use our validation tools to validate her data, which is important, especially if you're sharing your data with another researcher and you might mess up a data format accidentally, and so you can check it. And then also another brand new use case that we have where we're working with a geneticist to package data and extend our specifications with the stato ontology, which is statistics. So we're trying to come up with a more standard kind of like skeleton where you can write out your statistical tests for when you publish papers so that you can write your methods in a very reproducible way. So I think that summarizes everything, and I'm happy to answer any questions.
Okay. So let's see. All right, I have a question for you.
Sure.
So let me pull it up. So what are some of the data formats and data types that are currently supported, and which ones do you plan on supporting in the future?
Sure. So we mostly work with tabular data and, CSV, obviously. And so most of our tools and specifications are focused on tabular data, which is, what most researchers usually use, though sometimes it's Excel files instead of like a nice CSV file. We do not have a lot of good use cases for like imaging data right now. So, like fMRI studies, that's something that I think would be really interesting if we had better tooling for that.
Okay. So then we have one more, we have a few questions here. So raw genomics data sets can be on the order of hundreds of gigs to terabytes for a single experiment. It is recommended that such data sets are stored in the cloud and computed upon in place instead of downloading a copy. Do data packages support this compute in place model?
Yeah, so you can have the path to the data pack or to the data be in the cloud, or you can have it be a local file, either way.
Okay. And then last question I'm going to ask is, so do you have any plans for your data pipelining tools to work with other more general DAG tools like SnakeMake or Airflow?
I do not have those plans. That's a good question, and that would be really interesting. So we, open source project, we rely on our users to give us feedback and to give us suggestions for things that they would find useful. So if that would help you, that would be a really good user story for us to capture so that we could, figure out if other people are interested in doing that too.
All right, thank you.
Great, thank you.
So our next speaker is, so the talk that he gave was high dimensional data visualization with clustergrammar 2, Nicholas Fernandez. So you can give you a little overview of his talk.
Sure, thanks. How can I'll try and share screen real quick. Let's see. Okay, can you all see my screen? Or no? Okay, cool. Yeah, hi, thanks for having me. So yeah, we were discussing visualizing high dimensional biological data using clustergrammar 2. So it's a Jupyter widget that can be thought of as a data frame visualization tool. So you can basically load a data frame of data and perform hierarchical clustering, visualize high dimensional data such as gene expression data. And we'll just quickly go over an example or two. So here's some spatial gene expression data where we have this data is from 10x Genomics. So they basically are measuring a mouse brain over here. So you have individual spots where you're measuring lots of gene expression, basically like 30,000 genes. Here we've taken that data, we're looking at each spot as a column and all the most variable genes as rows. So you can like zoom into the gene expression data, and then we can like interact with the heat map and like select a particular gene, see the expression of that gene in the brain. And then we can also take this view and transform it to like a UMAP view. So you could basically see how dimensionality reduction interacts with your hierarchical cluster data. So you can see like a cluster here, you can select it, see where that cluster falls in the dimensionality reduced space. And in this case, it's this very interesting data where we actually physically know where these spots are, where these little sets of cells are in an actual mouse brain. So it's a fun data set to work with. But The way we normally use it is in Jupyter Lab or Jupyter Notebook, and we'll load a data frame of data. Here we have some protein measurements in about 7,000 cells. We can zoom in and see specific cells as columns, and here we're measuring about 14 protein markers. This is an assay called SciPy. So it's very interactive. You can reorder. You can also see the corresponding gene expression data for this data set, and then we can go in. The last thing I'll just briefly show is just labeling a particular cell type. So you can actually go in, use the dendrogram to annotate your data, and then those annotations are shared between different views of your data. So anyway, thanks for having me, and we can go ahead and answer questions.
Okay, so we have one question. So this is from Sebastian. Hello, this is a question for Nicholas Burnett. Okay, thanks for the great talk. I'm looking forward to using your methods on my community sequencing data. I have a couple questions. One, is there a minimum number of data needed for good statistics? And two, is there a metric like explained variance ratios in PCA that tells us how good the clustering is?
So the first question, I would say it depends on the statistical test you're doing. So if you're trying to do like differential expression analysis, for instance, then the more data points you have, the more robust your statistics are going to be. We also have some in the front end, some stuff where you can compare the clusters and you compare your prior knowledge and see how the relationship between if your cluster is enriched for a particular category you have. So the larger the data points, the better. But it depends on the nature of your data. It's very circumstantial. But I would say just even if you have a small data set, it doesn't hurt to just perform some sort of clustering and see what you see and just be able to play around with the data. And then in terms of the variance question, it also depends a lot on what you're looking for. So a lot of times what we'll do is we'll rank our dimensions by variance, and then we'll perform different filterings, and we'll see how that affects the structures in our data. So if we include like too many genes, like 1,000 genes or 2,000 genes, everything kind of gets too noisy. You lose a lot of the structure in your data. But if you start to whittle down the variance, and it can be very contextual also. So hopefully that answers those questions.
All right. Thank you. So it looks like I might have one more question. So is there, do you guys, can we use a custom dimensionality reduction algorithm, or are these limited to what's built in already into the library?
It's pretty modular. So one of the issues is that if you do dimensionality reduction, usually your reduced dimensions are kind of hard to interpret. So one thing I think maybe they're referring to the pairing of the dimensionality reduced view with the heat map. So yeah, any pairing you want. So like what we showed with that spatial data set, like we know the actual locations of these little groups of cells, and then we can also do them in different dimensionality reduced spaces. So it's pretty modular. But we're still cleaning up the API, so if you have any more specific questions. Yeah.
Great, thank you.
Cool, thanks.
So our next talk is called Interactive Image Processing for the Life Sciences with Scikit Image, Plotly, and Dash. And our speaker is Emmanuel Goulart. I'm sorry if I butchered your pronunciation. Here.
Yeah, SciPy. So my talk is about interactive image processing. And so it's in the line of the brilliant keynote by Anne that we had just before. And so the purpose of the talk is to show how to use some Lego bricks to build interactive image processing apps like the one I'm going to show you when I share my screen. So let's say that you have an image like this and you would like to build a machine learning model to perform segmentation like what you can do in Cell Profiler. So you can draw annotations using the Plotly library, and then I will explain just after how it's made, but just to show you the kind of interactivity we're looking for. And at the end, you will want to segment this picture. So the way it works is that this app combines several libraries of the scientific Python ecosystem. Here, it uses a Plotly graphing library for drawing annotations. It uses scikit-image to compute local features of every pixel under the annotations. It uses scikit-learn to train a random forest model and predict the classes of enabled pixels. And the UI is provided by Dash, which is a framework for analytical apps. So all these packages are used in a very modular way. I can give you a very short introduction to these different tools. So in particular, Dash is a web framework for analytical apps, but in pure Python. And this is what a typical Dash app code would like. So it's a Python script, which you run with Python myscript.py, and you define some elements in the layout. And so all the different elements here in the UI, the sliders, the checklist, and so on, would be one line of a definition. And after you define callbacks, which are functions called when one element, one of your components was changed, and to say what should be done to the other components. So it's a very modular framework, and you can use it to build quite powerful app, because in your callbacks you can use Python code, and so you can call scikit-image, which was the other focus of my talk, which is a collection of libraries of algorithms, to perform image processing operations. And we have quite a large variety of image processing operations, which you can take a look at the gallery of examples to see what we can do. We can compute local features. We have a lot of segmentation algorithms also. Another demo of my talk was using segmentation. And so I'd rather stop here and take questions.
Okay. So I know that I definitely had one question relating. I know I use scikit-image, Plotly, and Dash a lot.
Cool.
They're very nice tools, in my opinion. What are some features that you plan on implementing in those libraries, sometime in the near future, that might be helpful to our audience?
In terms of new features, you mean?
Yes.
Yes. So lots of them. So for scikit-image, what we plan is, first we're working on a version 1.0 after 10 years of existence. So we plan really to stabilize the API, also to have really 99% of functions which are compatible with 3D images. One major feature of scikit-image is that it's both 2D and 3D images, which covers most modalities of scientific imaging, but a few functions are not compatible with 3D imaging, so we'd like more 3D support. We would like also to have more registration functions. We have a big effort in terms of registration algorithms. We're also implementing type annotations in order to have more an easier compatibility with viewing and annotation tools like Plotly or Nafari. So that's for scikit-image, but we also plan to work on accelerating algorithms, which is sometimes a pain point which users have. So for Dash, So in terms of image annotation, we would like to develop integrated components for Dash, where let's say you could have like two figures with images corresponding to different modalities, and to have callbacks which are already predefined for your two images, two figures, or for example, having a 3D viewer, which is inside Dash, where you would have like the slider and the figure in order to navigate through the 3D volume. So for Dash and image processing, it's in the direction of having integrated components. And for Plotly, so we already have a number of drawing annotation modes, like rectangles, open and closed bar, circles, but we would like to add just simple points and polylines. And also to have a very fast image mode where you can load in a very fast way, let's say 4,000 by 4,000 images. So that's on the roadmap and coming soon.
All right, thank you. So we just had one community question, and then I'll let you go. So what limits are there on images that can be processed and image set sizes? And does the model suggest segmentation on images as you go?
So what was the last part? Sorry, what's the model?
So does the model suggest segmentation on images as you go?
For the app I just showed.
Yes.
Yeah. So for the app I just showed, you can indeed draw your first annotations and then have the segmentation, and then draw new annotations and have it in a live segmentation mode as Elastic would do, for example. And in terms of the limits of the data set, it depends a bit on your segmentation strategy, I would say, because if what you would, so I suppose that you're asking about the app I just showed, but if you have a very large data set which is composed of several images of the same kind, it's possible to train the classifier on one first image and then to batch process all the rest of images, which can be done in a non-live way, just a non-interactive way. So you can do this later on. So that's one limitation, which is not really a limitation, because you don't need to be there while the classification is done. If you have a very, very large image and you need to process it at once, then it's a question of how long you're willing to wait while the classification is done. And for this, we're using parallelization, because, for example, the random forest algorithm from scikit-learn is heavily parallelized. It's a forest of trees, and so it's quite easy. Well, quite easy. They do it in a very smart way, but you can just imagine that you have one thread corresponding to one tree. So this is parallelized easily. And for the computation of features, since we have several Gaussian kernels, we can also parallelize over this. So as long as your data fits in memory, it's the limitation is the time you're willing to wait. And if your data do not fit in memory, then you have third-party tools like Dask, for example, or tools which allow you to load data arrays which do not fit in memory.
All right, thank you. Thank you very much. So
you're welcome.
Move on to our next talk. So this one was called Making Sense of Clinical Data, Dimension Reduction, Data Visualization, and Clustering, Define Biological Mechanism. So speaker is Raha. So take it away.
Okay, so my talk, it sort of touched on two topics with an implemented example. So it was about working with clinicians coming from an engineering or data science perspective and why there might be communication issues there, what the different mindsets are, and then things to take into consideration when making models, thinking of them as being hypothesis generating, and erring on the side of simpler models that can be more easily interpreted, because if they're not easily interpretable, they're not as clinically applicable. And the importance, especially of your problem statement or definition and what it is that you're actually interrogating. And then the implemented example that I gave is sort of like a preliminary example of research that we're working on now of people with HIV who experience cognitive dysfunction over time. And so I chose the most simple model, which was a linear model of only 2 time points, before it even becomes predictive of why people are getting worse or better, and creating clustered models of groups of people that can then be interrogated separately. And then the next step is to figure out why, come up with a hypothesis there, and also to create longer nonlinear models. So if you guys have any questions, I'd be happy to touch on either topic.
Okay, so I had one question. So you had made a point about difficulty in interpretability, which kind of like balancing those is very important, which I thought was a good point. But maybe the way that we're analyzing our data, because a lot of your data analysis, I know that coming from multivariate hypothesis testing, it's kind of my background. So a lot of the tests that you were using were like multivariate t-tests, which have like a lot of underlying assumptions. So maybe the way that we're analyzing the data might not elucidate some underlying relationships. What can you comment about that?
Sort of, I think they might be separate things, right? So if you do like a t-test, you are assuming a Gaussian distribution, which if it's not true, maybe that's not the right test for you and you go for a different one. But when it comes to machine learning models, if you think of even the simplest one that hopefully would work with clinical data but barely ever does, classification trees are inherently nonlinear. So you don't have that assumption. And then as you go along, there are other models where they might have assumptions theoretically, but then empirically they don't apply as much. But even with dimension reduction, like PCA, I don't know if you call it an assumption, but you're saying that you're distinguishing based on variance. And so if your variables, if your measures are on completely different units, then you need to scale first. So scaling would be what you would do to meet the assumption that you would need for that technique.
Okay, thank you. So let's move on, I guess, to our next speaker. So our next, actually his battery ran out, it looks like. So I'll come back to him later. So our next speaker is, his talk was called Why Design Abstraction Matters for Analytics. Oh. Nope, he's here. Never mind. Let me invite him back.
You want to kick me out, and then I think I'm supposed to be the last one. So
yeah, I'll just minimize you. That's fine. Okay, so I'm sorry about that. So our next talk is called Molecular Infrastructure for Modeling Full Viruses with Pythonic Mediated Packages. Our speaker is Horatio Vargas Guzman. So take it away.
Hello. Thanks, Sambit, for the introduction. Yes, what we are doing is trying to democratize a little bit the modeling of viral capsids first, and then going towards to the whole virus modeling. As we have seen in this pandemic time, that it's very important to have access to information about new structures of viruses, for example, SARS-CoV-2, the one that originates the disease of COVID. And there was a huge scientific movement trying to update those structures and the molecular dynamic simulations of those structures rapidly in order to combat the pandemic. And those were shared. However, there was no unique platform like unique simulation protocol where we could clearly see what's been done in the simulations and the analysis of those structures. In particular, there was a controversy on the binding affinities on, for example, the spike proteins of the SARS-CoV-2 compared to the SARS-CoV 2002 to the AC2 enzyme of the cell, where many experimental researchers found a big difference in binding affinities, and that's how they explain the rapid spread of SARS-CoV-2, COVID-19, in comparison to the SARS-CoV 2002. However, other experiments show that the difference is not as big. And if we have a molecular infrastructure to analyze simulations rapidly, the free energies, for example, of binding of those simulations, and we can easily quantify those structures coming from experiments of active rays or cryo-EM and so on, then we can easily compare and maybe help interpret as well experiments, or even find new leads that will help us to combat, in this case, this huge pandemic that we are going through right now. However, the platform should work for any kind of biomolecule, any macromolecular assembly, and yeah, that's what Pi for All it's for. It's trying to democratize the access through these shared molecular trajectories of different kind of macromolecules. And democratizing mean because MOLSI and other infrastructures gathering a lot of scientists doing simulations, what they did is they are sharing their trajectories on different platforms, and we can obtain the trajectories and analyze them in a way that can be done by any scientist all over the world by using just a workstation with the GPU, and even in the cloud computing with the GPU, as I said. And that's the idea of Pi for All. If you are interested, just you can visit us on our GitHub page, or write us for any comment or any curiosity you may have.
Great, thank you. So I had one question. So I looked through your GitHub repo. I was wondering if you're planning on structuring the code that you currently have into a package structure, and then if you have any plans for that, and what kind of API would you kind of model after? Like, I know scikit-learn API is very well structured and a good place to start. So I was wondering what your plans are related to that.
Yes, that's exactly that's a very good question. Actually, we have I mean, the library it's ongoing. We have been creating this library while attending as I said, the simulations of different viral structures and so on that appear with pandemia. So the library is in a first stage now. We are, of course, we have in mind to follow the best practices in API development, and that's why also I'm here. I mean, to find those best practices and also contributors to our endeavor.
All right, thank you very much. So I'm going to move on to our last talk, after which we should have some time for a little bit of a moderated discussion. So our next talk is called Why Design Abstraction Matters for Analytics Tools, Accessible Neuroimaging Analysis with Neural Learning Tools. And our speaker is Ashwin Jolly. So take it away.
Alrighty. Can you see my screen and hear me all right?
Yes.
Cool. Okay, so my name is Eshin. I'm a postdoctoral fellow at Dartmouth College, and my training is actually in psychology and neuroscience. And the gist behind my talk was essentially, how might we think about design abstraction in terms of research-oriented software if we were sort of approaching it from a person-centric perspective rather than, let's say, an optimization-centric perspective. And so I sort of opened my talk a little facetiously saying, how would a bunch of social scientists design an analysis tool? And I tried to sort of summarize, I think, several key principles and ideas that you might think about when you're developing your own tools and building your own APIs. And I use the example of a library that I've been working on and I'm a contributor to that sort of integrates some of these things. And so one of those things is this idea of how much a piece of software can help scaffold your learning. And I tried to bring this example from developmental psychology, where there's this idea when you're in a classroom and you're teaching a student that you might be able to get them to optimally learn and acquire new knowledge if you keep them in something that some psychologists call a zone of proximal development. This idea of things are just out of reach, but there's some sort of assistance going on. So you can sort of build a mental model for, okay, I wonder what would happen if I do this, and then you can sort of test these predictions and ideas out. And we sort of try to think about things from this perspective when building our library. So there's some of the things that we do. The ideas we sort of try to get at here is this idea of coding at the speed of thought, which is a phrase used in different areas of software development. But the idea for us was, how do we minimize the distance between what you want to do and your goal, your intended actions in sort of natural language, and minimizing the distance between that and how you might express those ideas in code. And this is just one example from the slides on how you might try to do something like that. Now, at the same time, by facilitating this sort of expressive API design, you also need to think about whether you're hiding too much of the heavy lifting behind the scenes, right? And you don't want to abstract away understanding. And so one way to sort of think about this is that you might have some software that's designed that's very simple to use. Maybe it relies on these sort of GUI operations. This is a screenshot of SPSS, if people are familiar with that, versus sort of low level programming languages like Fortran, which you'll gain a deep understanding for something, but again, you may have to sort of operate at a level that's a little bit different from, I want to ask this scientific question. And so from our perspective, a sort of maybe missing axis of this layout here is the answer to this question: how much does a software tool assist you in your own self-directed learning? And I think that was one of the guiding principles for the design of our library, and maybe something that we might think about as a community when we're designing research-oriented software. So one of the ways to do this is to take that same tool, object that I described to you before and just sort of break down how we've built it. So this is just an example. Rather than sort of operating on a inheritance-based model, we actually used compositionality here. So because you already know how to use a lot of existing tools and we borrow and build off of them in the scientific Python ecosystem, you can bring some intuitions and ideas and play with some things already and therefore grow your knowledge in this way. I make reference to scikit-learn here because this is a quote someone told me when I was getting started learning Python and learning statistics and machine learning. And I think one of the, scikit-learn is very popular as a lovely API, but I think that the real beauty from my perspective is that it really helps you build an intuition, a conceptual understanding, if you sort of work with it and use it, right? So what I mean by this is that the way that estimators and things are designed, you sort of are getting a bunch of conceptual knowledge for free. You're understanding what algorithms algorithms may be doing, you're understanding some sort of foundational concepts. And as a result, using scikit-learn can actually make the rest of machine learning available to you, right? It sort of provides you some sort of conceptual foundation. And I think that's just something that we can think about when we're designing research software. So those are the key sort of points that I made in the talk. These were the sort of 4 key ideas that I tried to touch upon. And I think it's maybe a little bit of a different, maybe controversial way to sort of design libraries. I'm not saying optimization and good code design in sort of like the classic practices are we should do away with them. I'm just sort of thinking about how we might use tools as a way to encourage people to sort of learn and grow their own understanding in this sort of assistive way. And just as a sort of a little bit of proof in the pudding, we have, this is the documentation site. We have some examples so you can learn and play with some of this stuff. We also have an open textbook. This is actually a real class taught at Dartmouth College in New Hampshire in the US. If you're interested in learning about neuroimaging and the basics, a lot of the tooling in this class is it's completely open and was taught using this library. And the library actually powers this web-based interface that is currently under development, where you can sort of get a very quick way to sort of prototype some imaging analyses and do some machine learning for functional imaging analysis just through your web browser. So that was the basic sort of gist. Happy to sort of answer any questions or explain anything further.
Okay, so we have one question. So can you say more about the process of developing the API so it let people code closer to natural language? And is it the result of many iterations? How do you see it evolving?
Yeah, that's a really good question. It's definitely a process of iteration, right? Because we didn't sort of sketch this out ahead of time and then say, okay, this is how we're going to do it. I think in much the same way you might approach teaching students, or if you're a parent and you're raising a child, you might, if we're honest with ourselves, we may not know the best way to sort of engage with them. Part of it is to sort of give it take to see sort of what gels and what's intuitive. And I think it's something working for us in the development of this library is that I do neuroimaging analyses myself, and I teach them and I mentor a lot of students. And so part of the goal is how can I sort of take something that might seem very complicated. I want to create a brain map with sort of the activity of what's happening in your head. And how can I take somebody who has maybe some very, very basic foundation in statistics, like running a t-test or computing a mean, and sort of get them to that stage in this sort of efficient way, right? And one way to do that, we sort of approach this is how much you express your goal in natural language, and how can we design an API library that's almost as close to that natural language as possible, while still not sort of obscuring anything in terms of your understanding or any of the sort of core operations that are happening. All that should be accessible, this idea of a glass house, but it also should be intuitive. You should be able to sort of say, oh, I want to do it this way. I wonder if this would work. And more often than not, you'll learn by realizing that your intuitions are correct, and the library sort of rewarding you for doing something like that.
Okay, and then last question was, your presentation was engaging and visually appealing. How did you prepare your slide deck?
I'm stuck in the Apple ecosystem, and for a long time ago, I ended up using Keynote, which is the Apple competitor to PowerPoint. It's free to use. I tend to like it, so that's pretty much where it comes from. I also just want to say thanks to everybody transitioning us to the online format. This is actually my first SciPy, and it's been super awesome. The community is so welcoming, and it's so great to sort of meet all these folks and stuff. So, let's keep that up. It's awesome.
All right, thank you. So right now would have been time for moderated discussion, but I think we're kind of getting close to the end of time. But I noticed a lot of questions about cluster grammar. So I think that I'm going to invite him up just for a little bit, and then we can call it a day. So let me see.
Hi, thanks.
Hi. So one question I think that's kind of important is, does the cluster grammar API have hooks or callbacks that let me do cross-filtering with other widgets or plots?
Yeah. So we have callback functions that can be run on certain like click events or certain mouse events, but we're still developing the API on that. So if you have any specific type of interactions that you want, then you just let us know. We can help you with that. But we're generally using the widgets traitlet thing to kind of like sync back and forth between the front and the back end.
All right. And then another question was that I have a data set with 250,000 rows and 1,000 columns. Will clustergrammer work with that?
Yeah, so the 1,000 columns will work, but the maximum number of rows or columns that we can handle in the current version is around like 10 to 20,000 or so. Because the approach we're taking is we want to have very fast interaction, so we're loading everything to the front end. And later on, we'll work on like when you zoom in, you'll bring in more data. But at the current state, when we have a data set of that of around like hundreds of thousands of data points, we'll usually try and run like some either subsample or we'll do some kind of type of downsampling. So maybe we'll produce 5,000 clusters to represent that data set and then visualize that, and that's an approach.
Okay, and then I think the last question I'm going to ask you is, have you implemented clustergrammer on longitudinal data?
Yeah, so recently we did a little bit with, I was looking at the New York Times COVID data. So they have, I'm just sharing my screen right here. We have this heat map of like COVID data spikes, and we have like every county in the US, and then we have time across the y-axis. And then you can like zoom into like sections. You can like click here, and then it'll highlight like which counties are in that cluster. So we've a little bit of experimentation with it, not something huge. And then this just highlights that you can take the software and you can run it on observable, the JavaScript component of it. But the fun part is it's Python on the back end, it's using GitHub. And but yeah, if you have any specific questions, we can discuss.
Okay, so I think that about wraps it up. Thanks everyone. Thank you for the speakers for being here and telling us about your interesting research. And I guess that ends it.